Okay, so great. So I'm very excited to start this session, which is on quantum-enabled high-throughput sensing. My name is Amir Naeini. I'm an associate professor in the applied physics department, and I first want to sort of thank everyone, co-organizer Peter Maurer, the QFarm team led by Juna, QFarm for the funding, the Moore Foundation for a lot of the funding that lets us have this beautiful space and food, and all the speakers for coming very long distances or not so long distances. So thank you, everyone. I also want to thank the team in my group who's been working on a lot of the things I'm going to talk about, in particular Oliver Hitchcock, Matthew McSnovich, and Kaveh Pezishki at Stanford, and Mert Yüksel from Caltech, who's in the audience here. This is a close collaboration with Michael Rukas' team at Caltech. And we also work on the theory side with Mark Dickman at MSU and Rob Phillips, also at Caltech. So for the past several decades, one of the most exciting frontiers of biological measurement has been working with single molecules, and this is one of the things that W has pioneered, and it's really changed our perspective on what's possible, optically detecting and studying individual molecules in very complex environments. And now we heard from Peter about how these qubits can be actually brought into the protein, so the protein can be the qubit. And this capability allows us to remove this averaging issue, ensemble averaging issue. Now there's the other dimension, which is equally important, which we heard Steve Quake talk about and pioneered. He pioneered the tools for kind of high throughput measurements, going to the scale of entire organisms, developing cell atlases by combining microfluidics and very rapid DNA sequencing. So looking at these two frontiers, and these frontiers are not completely independent, but we can think about sort of the juxtaposition of these two. Can we kind of bridge these two frontiers and make something that's really provides you the depth of that kind of single molecule richness, plus the scale and throughput that allows you to work at very large scale. One particular problem that's very important is identifying individual proteins. So it is likely that we can't amplify proteins the same way we can amplify DNA and RNA. And so every single protein needs to be identified individually. And this poses a challenge because you end up needing extremely sensitive detectors that have enormous dynamic range. So we're talking about range and mass that's like 5 orders of magnitude. And they have to operate with very high throughput, and they have to detect proteins that can, again, be very rare or extremely common, with sort of ranging concentration that scales like 7 orders of magnitude. So if you look at the proteins in a mouse cell, there's a histogram of the types of proteins and the number of copies that there are in each. So current technology, you can get to sensitivities of around 10 to the minus 6. And that means that we can detect the proteins that are sort of on the right side of this graph. And so on a single cell, a lot of this proteome ends up being dark if we're detecting single proteins. So so that's a sensitivity issue, and then there's a throughput issue. So in a typical mammalian cell, there might be a few billion proteins. If you look at the sort of state-of-the-art mass spectroscopy system, the rate at which it works, it would take a year to, even if it had that sensitivity, to detect all of these proteins. Now, one kind of surprising avenue, I think, for trying to address some of these problems and challenges is emerging from the field of quantum computing. There's this global effort to build quantum computers, and these fundamentally bring new types of tools and concepts and instrumentation to us in this field, people have now developed extremely low noise materials, which are important if you're trying to build sensors that are very precise, very broadband amplifiers for reducing the noise and getting to sort of single molecule signal excitation sensitivities, single photon sensitivities in their case. There's now demonstrations of single site, single shot readout over large scale. This is an example from people trying to build neutral atom-based quantum computers. So trapping and addressing hundreds of thousands of atoms at single site individually and moving them around and sending short pulses to them is becoming possible now. And kind of more generally, we have the ability to do very high dimensional, extremely fast control. So this is a picture from inside the one of Google's quantum machines. This is a 100-qubit system, and you can see thousands of cables. These are all broadband signals that are being in, and there's a whole infrastructure for real-time feedback control that's being developed for this application. So there's also developments in new types of memory elements and sensors that work in conjunction with these quantum technologies. And one type of sensor that we work a lot in my group are mechanical devices. I'm very excited about these. So, Peter showed that mechanical sensors don't make good clocks. It's true, they don't, and that's because they're good sensors. That's the So we can, detect energy at single phonon excitations. We've been able to demonstrate that now. There's been work showing that you can make the best accelerometers, the best sensors of force. There's, of course, the demonstration by LIGO of gravitational wave detection on a much larger scale. People have been able to do very sensitive temperature measurements. This is important for example, detecting single molecules through thermal optomechanical measurements. And what I'll talk about are inertial mass sensors, which are sensors that work by detecting changes in mass. So just to recap, if we want to look at a single proteome, a single cell proteome, we need to, in kind of a reasonable amount of time, we need to basically detect 10 million proteins per second. And we need single Dalton, proton level mass sensitivity on each of these proteins to be able to distinguish them. So what we proposed, and this was funded by the Moore Foundation through an Inventor Fellow, was a large array of mechanical devices on a chip. And what makes these mechanical devices special is that first, they are very small. So the kind of active area where you see that vibration is much less than a cubic micron. And that means that the mass is very small, and so they're sensitive. The other aspect that's important is that we can make hundreds of thousands of these on a chip. And the third aspect is that we can address all of them in a very simple way. So we don't need to have a whole measurement chain for each of these resonators individually. In fact, we can just basically hooking up a commercial vector network analyzer to this chip, we can see a large number of these mechanical resonators and track them in parallel by doing frequency multiplexing. So this is what these devices look under a microscope. And so the nanoscale provides sensitivity, and high bandwidth we get from frequency multiplexing. And so what the vision was to have basically 100 pixel arrays for every RF line. We need about 100,000 of these, so we're talking about 1,000 RF cables, and that's exactly what the current quantum superconducting quantum machines. That's what their infrastructure is based on. So just to go through the basics of mass sensing, the physics is quite simple. So we have a mechanical resonator. It has a certain mass. Something lands on it, so the mass changes, and so that leads to a change in frequency. We have to measure this frequency. So our ability to determine the frequency of something is given by it's kind of a fractional frequency uncertainty or Allen deviation. And this imprecision in frequency leads to an imprecision in the mass. So if we have something that has a mass of 100 femtograms, which is roughly what these mechanical resonators are at, we need to have an Allen deviation less than 10 to the minus 10 to be able to detect that. So that means for our devices, we need roughly about 1 hertz. We have to see frequency fluctuations on the order of a hertz. A sort of more kind of quantum analysis of how this frequency measurement works. It's also classical analysis, but we send in a signal, a coherent signal, let's say from a electronic device into the resonator. We get a reflected signal back. A mass lands on the resonator. This leads to a frequency shift. And then we detect this frequency shift by looking at the change in the phase of the reflected RF signal. So we can integrate the signal over some integration time tau. And of course, we have to integrate the noise over that time as well. And if we look at what the SNR is, the SNR is this last equation on the bottom right, which shows that it scales as 1 over square root of time, the integration time, and 1 over square root of the number of photons that are used to drive this thing. So we can plug in the best numbers that we have for the mechanical Q of these devices. This is where we were a few years ago, 10 to the 5. Integration time 10 milliseconds. So that means 100 proteins in a second per device. Of course, we have to get to 10 million, but we get that by scaling to 100,000 of these devices, hopefully. And given the mass, we can find, to detect 1 Dalton, we need about 10 million photons, and this is a completely reasonable power level. So, the basic physics works out. So we were very excited. When I wrote this proposal, the state-of-the-art demonstration was from Michael Rucas's group showing mass sensitivity on the order of 2 Dalton. And our plan was we were going to get to less than a Dalton sensitivity very quickly until, over a couple of years. Of course, when we tried this out, it didn't quite work, but for interesting reasons. So we were able to measure proteins. This is data taken by Mert, Oliver, and Matthew and Kaveh a little while ago, showing that, for example, it is data taken at Caltech with our devices showing apoferritin jumps in the mechanical resonance frequency. But you can see that in addition to the jumps, there's a lot of variance in the frequency readout there. And so our measured sensitivity was much worse than the 1 Dalton. It was maybe a few orders of magnitude worse. And so we spent a bunch of time trying to understand what's causing this excess frequency noise. In fact, it was a kind of surprising result. You would expect that when you go to lower temperature, the system cools down, the frequency noise should actually go down, but we saw an increase in frequency noise as we went to lower temperatures. We understood this through dynamics of coupling between intrinsic defects in the structure, and now we've actually really understood these defects to the point where we can even couple strongly to individual defects. So this is a vacuum Rabi splitting from our mechanical system, coupling to an individual defect, and we can see strong coupling there. So it's entanglement between our mechanical system and the defect. This is still work in progress. The data I showed you was with the our devices from a couple of years ago. Over time, we've been improving these quality factors, and now we're starting to get into a regime where these devices hopefully can get to the a few order of magnitude increase in sensitivity. We're also building setups now to combine the commercial mass spectroscopy system, for example, Orbitrap system, with a dilution fridge with the infrastructure of that's common in superconducting qubit labs. And we just commissioned a fridge specifically for this purpose a few weeks ago. So that's all I have to show for stuff for my group. But I just want to say that this approach for scaling the systems up by lithographically defining arrays and reading them out and kind of multiplex quantum hardware, this is a powerful strategy. And I think this is one way of getting to higher throughput. And for the rest of the session, we're going to hear about different ways of increasing throughput. So the first speaker, Jen Dion, is going to introduce us to scaling by increasing the density of photonic devices. And the question is, what if we build a sensor that basically where every one of these it's composed of many thousands of devices, and every one of these can do complex multi-omic measurements in parallel. The second speaker is Ashok Joy from University of California, Berkeley, and here the scaling is by multiplexing in time. So if we, instead of building a large static array, what if we have these kind of cheap sensors and we parallelize measurements in time domain? So finally, I just want to also take a moment to talk about kind of fundamentally new types of measurements that can be enabled by these quantum technologies. So one of these that I'm very excited about are phonon counting measurements. The way we usually measure phonons is by measuring voltage fluctuations. That's how a microphone works. But you can also there's a quantum system. Instead of asking it what is the amplitude of the vibration, you can ask it what is the energy, and then you get fundamentally different information that's not accessible by a standard microphone. And a dream for us to do an experiment where we would take one of these nanomechanical devices, that we get a protein on it, the way we've been able to do with apoferrin, for example, and then we pump, for example, a known mid-IR transition, one of, let's say, the CH or CO bonds in the protein. So we dump some energy into it, and then we just listen. So that we know that that excitation will relax through a number of phonon pathways, generating phonons 30, 150 gigahertz, and we can hopefully directly measure those phonons. These superconducting quantum devices make fantastic tools for measuring phonons. That's one of the key causes of decoherence in superconducting qubits. So we're thinking of how to use that, and that's something I'm very excited about. So. And finally, I just want to thank the sponsors. Initially, this work was funded by National Science Foundation, and we've received a significant amount of funding from the Moore Foundation and AWS for improving these materials and understanding the physics of them. So I guess, thanks.